I'm not clicking. You're clicking. Also, I need to read some papers and watch a video, so I'm going to do that, I think. Also, I need to sneeze. Excuse me. Oh wait, that's right. If I'm gonna watch a video, I cannot
I'm going plasticity.
have music go.
Transcription and translation. So, in early work from the mid-1980s, a collaboration between Eric Kandel and Sam Shacker explored long-term facilitation in cultured synapses, that is,
Wait, I'm not ready.
synapses between dissociated sensory neurons and motor neurons from hemiplegia, which in co-culture form synaptic connections. So here you see the presynaptic sensory neuron firing action potential and the postsynaptic motor neuron called L7 with a synaptic potential in response to this presynaptic action potential. And they tested what the role of protein synthesis in this long-term plasticity would be. And today, of course, we accept that protein synthesis is critical for long-term synaptic plasticity, but the situation was somewhat murkier in the mid-1980s. In the early 1960s, Lou Flexner at Penn.
Actually, wait a minute before I finish this. Reminder, this is the thing at noon. Gross.
And done experiments on rats, feeding them antibiotics that block protein synthesis, and found that those animals had an impairment in long-term memory. However, the animals fed high concentrations of antibiotic were fairly sickly, and it was hardly clear that this was the only alternative, this is the only explanation, and certainly an alternative explanation would be that there were very nonspecific effects of this high level of antibiotics. So, leaving the animals behind and studying cells in culture, sensory neurons and motor neurons in co-culture, Sam Shacker and Eric Kandel and their colleagues were able to re-explore this issue. So what you can see is, in the first experiment shown here on the left, if you give one pulse of serotonin by itself, that's insufficient to produce long-term synaptic facility.
Wait, what? I was listening to that.
Hello, this is a synaptic motor neuron called L7 with a synaptic potential critical for long-term synaptic plasticity. But the situation was somewhat murkier in the mid-1980s. In the early 1960s,
oh wait, do I have to actually download the whole thing? God damn it. I didn't realize it was only 15 minutes. Hell, how long is this actually? It was only 15 minutes, but it's not. Don't be an hour, don't be an hour, don't be an
Hello,
hour.
this is Tom Abrams.
You have no idea what's going on in this paper. Yeah, me neither. Let me see if I can suss this out.
Hello, this is Tom Abrams.
Hello, Tom.
In early work from the mid-1980s,
All right, hit me.
a collaboration between Eric Kandel and Sam Shacker explored long-term facilitation in cultured synapses, that is, synapses between dissociated sensory neurons and motor neurons from hemiplegia, which in co-culture form synaptic connections. So here you see the presynaptic sensory neuron firing an action potential and the postsynaptic motor neuron called L7 with a synaptic potential in response to this presynaptic action potential. And they tested what the role of protein synthesis in this long-term plasticity would be. And today, of course, we accept that protein synthesis is critical for long-term synaptic plasticity, but the situation was somewhat murkier in the mid-1980s. In the early 1960s, Lou Flexner at Penn had done experiments on rats, feeding them antibiotics that block protein synthesis, and found that those animals had an impairment in long-term memory. However, the animals fed high concentrations of antibiotic were fairly sickly, and it was hardly clear that this was the only alternative, this is the only explanation, and certainly an alternative explanation would be that there were very nonspecific effects of this high level of antibiotics. So, leaving the animals behind and studying cells in culture, sensory neurons and motor neurons in co-culture, Sam Shacker and Eric Kandel and their colleagues were able to re-explore this issue. So what you can see is, in the first experiment shown here on the left, if you give one pulse of serotonin by itself, that's insufficient to produce long-term synaptic facilitation, measured 24 hours. On the other hand, if you give a series of 5 pulses of serotonin, each lasting about 5 minutes at 20-minute intervals, and you come back then one day later, you see nice long-term synaptic facilitation.
All right.
And then in these experiments, they tested the role of anisomycin or emetine, both of which are protein synthesis inhibitors, or alpha-mannitine or actinomycin D, which are transcription inhibitors. And what you can see is, here's the average effect of 5 pulses of serotonin measured a day later. There's perhaps a 65 to 70% increase in synaptic strength. If they give, I don't remember what these stand for now. This is anisomycin. They block that increase completely. Emetine blocks the increase completely, so those are protein synthesis inhibitors. And then 2 transcription inhibitors have pretty much the same effect: actinomycin D and alpha-mannitine. So it seems that both protein synthesis and gene transcription are critical. And then, in an interesting set of experiments,
I think we have different definitions for the word interesting.
Time window at which protein synthesis was important. And I always find these results surprising.
I don't.
So here they inhibit protein synthesis with
Or maybe I do.
anisomycin, which is reversible several hours after washout. So they give protein synthesis for a 3-hour window, 12 hours to 9 hours before the start of training.
Okay.
And then in this protocol, they give the protein synthesis inhibitor during the training, starting an hour before and going up to the end of training. And in this group, they start the protein synthesis less than an hour after training. And you can see, whereas the protein synthesis inhibitor during the 5 pulses of serotonin completely blocked the long-term facilitation, here the effect of protein synthesis inhibitor is modest. And if they delay the protein synthesis application and they give it for a period of 3 hours, starting, it looks like 5.5 hours after the end of, or 6 hours after the end of serotonin, they have no effect at all. And even more dramatically on the right, if they delay the protein synthesis inhibitor for perhaps 40 minutes after the end of serotonin, and then they give the protein synthesis inhibitor continuously for 22 hours, there's no impairment of long-term facilitation, or at best a modest impairment. So this is really quite amazing. And the same is true if they give the transcriptional inhibitor alpha-mannitine.
Not really.
So, surprisingly, protein synthesis is not important in a continuous fashion for synaptic strength to build up, but rather it's important around the time of the serotonin exposure. And in these experiments, David Sweat, Eric Kandel, and Ari Barzilai and Tim Kennedy looked at the total protein synthesis occurring at different time points. And here, instead of using pulses of serotonin, they use continuous exposure to serotonin. And what you see is that initially during the serotonin exposure, there's a drop in protein synthesis, followed by an increase, then this decays, and then there's a late phase of protein synthesis. And this is very interesting because we would consider this to be an early phase, and this is a late phase. And notice the two are not connected with a continuous increase, but rather there's a decline in between.
Fascinating.
And then the experiments down below, which are quite interesting, they looked at what happens to to protein synthesis, depending on whether they inhibit gene transcription or protein synthesis, as over here. So the white open bars are the control. Again, it's really the same data as shown up here. There's an early decrease followed by a slightly later increase, still during the serotonin. And then there is this late increase at about 3 hours.
Okay.
If they block transcription, the early decrease and the early increase are unaffected, but the late increase at 3 hours is completely blocked. If they block protein synthesis, if they block transcription slightly later, then they only partially reduce the rise in protein synthesis at 3 hours.
Okay.
So these
it.
data suggest that gene transcription in this early window is important for the increase in protein synthesis here.
Yep.
What we can imagine is that there's a cascade of events, of molecular events, in which new gene products during the serotonin, transcribed during the serotonin, cause increased protein synthesis at a later time. So presumably, these include transcription factors synthesized here, expressed, and those transcription factors in turn turn on protein synthesis here, perhaps. And what you can see here is if they give anhysamycin early on, around the time of the serotonin, and the anhysamycin, although they're washing it out here, will presumably persist for a little while. And then they look at protein synthesis. The protein synthesis increase here is completely eliminated. That's consistent with the idea that the early gene products are perhaps regulators of transcription. And these transcription factors are responsible for this late phase of protein synthesis. So I'm going to move now to looking.
Do I use a hairbrush? Kinda? I don't know. It's like a brush with little pokey bits. What is a hairbrush, theoretically? Are you looking for one with, like, hair on it? I have a thing like this. Oh, wait, that one actually has hair in it. No, I don't. I don't have that. I didn't know they made hairbrushes with doodads in them. I have a thing like that. Sort of. I don't know if I should be using something else. I probably should. This one's got boar bristles. Cool. I haven't had long hair in 7 years and it's not growing in how you remember it. In what way? Is it growing? Like, I don't know. I don't know what that means. Is it like a shape or what? Or like your hair looks weird in comparison to mine? Or is that what you're saying? Oh yeah, that's the problem I've had. The secret is conditioner. Or else you will have just a poof mess.
At the role of the cyclic AMP pathway in this late phase or LTP, late phase long-term facilitation that occurs at 24 hours, and this is, as we just said, transcription translation dependent. When they started this work in 1988, that was not so clear. And in particular, it wasn't clear how cyclic AMP might be linked to protein synthesis. But what you can see here is these are a couple of examples, and instead of using serotonin, they use a cyclic AMP analog, and they pair it with IBMX, which reduces cyclic AMP dependent cyclic AMP degradation by phosphodiesterases. And what you can see here is, and it's not exactly clear, although we've used the same kinds of combinations in our lab, it's not clear why the cyclic AMP analog may not be as effective alone as when it's paired with IBMX. Even though these analogs are supposed to be phosphodiesterase resistant, it's possible there's some degradation. So here what you can see is that when you pair cyclic AMP, a cell permeant analog, with IBMX, you get facilitation 24 hours later. IBMX alone has no effect. And furthermore, this effect of cyclic AMP plus IBMX
about the
is
background
dependent
memory
on
board.
synthesis. So this begins to link the two stories. Now, before going to talk further about transcription factors, I want to mention early experiments by Jimmy Schwartz, who in the mid-'80s explored the idea that intermediate-term facilitation, perhaps long-term facilitation, depended on persistent activation of cyclic AMP-dependent protein kinase. And what he speculated was that there might be degradation of the regulatory subunit, which acts as a tonic brake on the catalytic subunit, so that even in the absence of cyclic AMP with a very modest rise of cyclic AMP, you could have a substantial activation of the catalytic subunit. And this is really a speculative cartoon that looks sort of like a model, but it's not a very quantitative model. But the idea is here, with the normal level of regulatory subunits, it takes a rise of cyclic AMP, say this much, to produce a certain amount of activation. If you have a degradation of some of the regulatory subunits, now there's sort of a persistent activity, and with a rise in cyclic AMP, you get more powerful activation. So it both gives a tonic level of activation of the cyclic AMP-dependent protein kinase catalytic subunit, and it enhances the activation that you get when serotonin comes around again. And they speculated.
Is the audio related? I don't know, dude.
If there's a regulatory subunit degradation produced by the early exposure to serotonin, then later exposure to serotonin are more effective. And this could be a reason why repeated stimuli during learning, repeated exposures to information you're trying to learn, are more effective when they're spaced out in time rather than massed together. We know that subjectively as students, but it's true in animal studies as well, that spaced training is generally more effective than massed training.
Wait, what was the reason for that?
So Steve Greenberg together, we know that when serotonin comes around again. And they speculated that if there's a regulatory subunit degradation produced by the early exposure to serotonin, then later exposure to serotonin are more effective. And this could be a reason why repeated stimuli during learning, repeated exposure to produce a certain amount of activation. If you have a degradation of some of the regulatory subunits, now there's sort of a persistent activity, and with a rise in cyclic AMP, you get more powerful activation. So it both gives a tonic level of activation of the cyclic AMP-dependent protein kinase catalytic subunit, and it enhances the activation that you get when serotonin.
This makes sense.
And they speculated that if there's a regulatory subunit degradation produced by the early exposure to serotonin, then later exposure to serotonin are more effective. And this could be a reason why repeated stimuli during learning, repeated exposures to information you're trying to learn, are more effective when they're spaced out in time rather than massed together. We know that.
I don't search images on Google Image Search.
But it's true in animal studies as well.
Usually unrelated.
Is generally more effective than massed.
It's also less interesting to look at a picture than an article.
So Steve Greenberg, working with Jimmy Schwartz and Hagen Bailey and Vince Castellucci, quantified regulatory subunits. And he did this in a gel in which he labeled regulatory subunits. And what they found was that 3 types of regulatory subunits, based on their molecular weight, were all reduced in amounts compared to controls, because the control would be 100%. So this one is reduced even more than a 50% reduction. This slightly larger molecular weight shows about a 30% reduction, and this largest one shows perhaps a 40% reduction. In contrast, there's no change in the amount of catalytic subunit. And somewhat surprisingly, this loss of each one is reduced even more than 50. Steve Greenberg, working with Jimmy Schwartz and Hagen Bailey and Vince Castellucci, quantified regulatory subunits.
Of what?
And he did this in a gel in which he labeled regulatory subunits.
Of what?
And what they found was that 3 types of regulatory subunits, based on their molecular weight, were all reduced in amounts compared to controls, because the control would be 100%. So this one is reduced even more than a 50% reduction. This slightly larger molecular weight shows about a 30% reduction, and this largest one shows perhaps a 40% reduction. In contrast, there's no change in the amount of catalytic.
In what?
Regulatory subunit degradation produced by the early exposure to serotonin. Then later exposure to serotonin are more effective, and this could be a reason why repeated stimuli during learning, repeated exposures to information you're trying to learn, are more effective when they're spaced out in time rather than massed together. We know that subjectively as students, but it's true in animal studies as well, that spaced training is generally more effective than massed training. So Steve Greenberg, working with Jimmy Schwartz and Hagen Bailey and Vince Castellucci, quantified regulatory subunits. And he did this in a gel in which he labeled regulatory subunits. And what they found was that 3 types of regulatory subunits, based on their molecular weight, were all reduced in amounts compared to controls, because the control would be 100%. So this one is reduced even more than a 50% reduction. This slightly larger molecular weight shows about a 30% reduction, and this largest one shows perhaps a 40% reduction. In contrast, there's no change in the amount of catalytic subunit. And somewhat surprisingly, this loss of pKa regulatory subunit actually required protein synthesis. So here is what happens with 5 minutes of serotonin exposure, and this is looked at shortly after the serotonin, not a day later. So this is the amount of regulatory subunit, I think a couple of hours after the onset of serotonin. And what you see here is that with 1 exposure to serotonin, there's no change. With 2 hours of serotonin, there is, which is equivalent to a series of pulses of serotonin, there's a substantial reduction. And if you give 2 hours of serotonin followed by 3 hours of anhysamycin, you still get a reduction in the amount of regulatory subunits, suggesting that there's a protein, well, as we'll see in a minute, there's a protein synthesis dependent, but the protein synthesis dependence is during the serotonin. Because if they give the serotonin here for 2 hours and during that time, and slightly before they give anhysamycin, now they completely block the decrease in regulatory subunits. And anhysamycin alone has no significant effect. So what's surprising here is that the loss of the regulatory subunit actually is a translation-dependent event. And at first glance, that's sort of surprising. So within a couple of years, Pramod Dash, working with Eric Kandel, had learned about CREB, which had been identified by Mike Greenberg in Brain as a transcription factor interacting with the cyclic AMP response element, an enhancer upstream of cyclic AMP regulated genes. And what Pramod Dash did here was he developed what he called it later a transcription factor decoy that might sop up all the activated CREBs. So this is basically a double-stranded oligonucleotide that corresponds to a mammalian gene that has a CRE. So in other words, it's the regulatory region of this mammalian gene. And when the concept is when this little double-stranded oligo is injected and CREB is phosphorylated, you can see the little phosphate group on the CREB. Now CREB, instead of binding to the normal CRE in this enhancer and turning on gene transcription, now the activated CREB will bind to these little decoys floating around in the cell. And so that was the hypothesis. And what you can see here is it actually worked pretty much that way. So Here, a control
Magic.
on top. You see long-term facilitation, very nice facilitation 24 hours later. With a control oligonucleotide, they see very nice long-term facilitation. With an oligo containing the CRE, they block long-term facilitation. But then, in those injected cells, if they come back 24 hours later and give serotonin, they can get nice short-term facilitation. And the group data are what you see here. The CRE containing oligo, double-stranded oligo, it was like 31, completely blocked the long-term facilitation. This was actually a mammalian gene, I think, the somatostatin CRE. And
Great.
all of these controls had no effect on the long-term facilitation measured 24 hours later. And of course, there are no effects on short-term facilitation. So the long-term facilitation depends on Krebs-initiated transcription. And Bong Kang looked at using a similar approach, but really the opposite. Instead of using decoys, he used a reporter. So here he has a series of CREs, 5 CREs from the VIP, vasoactive intestinal peptide
Preptide?
promoter, upstream from LAC-Z. So this VIP is a CRE or Krebs regulated gene, and using this construct, he can stain for cells that express beta-galactosidase. And what you can see here is that in the control, there's very little beta-gal expression. But here, with 5 pulses of serotonin, these are in sensory neurons, I should say, sensory neuron cluster, that have been injected with this construct. So here, a large number of sensory neurons express a high concentration of beta-gal. And what you can see here is
It's a
just
really bad
controls.
superhero
Here
name.
is the VIP promoter.
The Adventures of
that
Beta
we've seen here.
Gal.
And they're adding IBMX to potentiate the effects of serotonin. Here is without the serotonin, with the open bar, with the serotonin,
Yeah,
they
that's
get a
probably dumb.
nice increase in beta-gal staining. If they remove the CRE from the VIP promoter, there's no effect.
Oh, God.
This is a control
This is
promoter
so
and a second control promoter. And then
Of course, this is
here,
the professor
they
that
can
makes a
block
very
the
big
effect
point
using the same
of you
approach
describing every
as
single one of these figures in class.
if they inject the oligos containing the CRE. And then, interestingly, they looked at the requirements for repeated pulse of serotonin. What you can see is that there's a gradual increase in beta-gal expression. So with 1 pulse of serotonin, they get no increase. With significant increase. With 2 to 3 pulses, they get a small but significant increase. And then with 4 to 5 pulses, they get a much nicer increase in beta-gal staining. So again, as we talked about earlier, it seems like repeated pulses of serotonin are required for effective activation of this transcription cascade. So now I'm going to turn to experiments done slightly later, looking at the signaling involved in
Later again.
late phase long-term potentiation in hippocampus and CA1 pyramidal cells, looking at the Schaffer collateral or CA3 to CA1 pyramidal cell synapse. And in this paper, they're looking in the first figure at the contribution of 2 different protein kinases, CAMK2, which we already believe is important in long-term potentiation, and cyclic AMP-dependent protein kinase, or PKA. And they have KT5926 that they use to block PKA2, and KT5720 to block PKA. So with a single tetanus, you can see that LTP, within perhaps 20 minutes or less, is substantially reduced by inhibiting CAMK2.
Yep.
Whereas there's no effect of inhibiting PKA.
Okay.
And anhysamycin, which blocks translation, has no effect. So this is protein synthesis independent and PKA independent within a single tetanus. If, on
Right,
the
I was
other
reading
hand,
that. Go back.
they use a strong stimulus,
Blocking
And now
CAMK2. What was
notice
the other one?
that-
PK.
unlike the control, which is fading by 1 hour, which is down substantially about 1 hour, with a strong stimulus, they have more stable LTP going out to an hour and a half,
My magical girl identity
They-
can be Beta Gal.
eliminate
God.
this almost really completely by inhibiting CAMK2. But notice now PKA
Gal is such a
inhibition
dated
has some
term.
effect.
I don't know why.
And
Just
then
makes me
they
think of
looked
senior
at,
citizens.
or they actually had to develop these protocols to induce long-term or late phase. Of course, it's all long-term potentiation, but this is late phase or more stable LTP in the slice. So here they use 3 trains. And you can see that they have stable LTP out at 3 hours. And in those early days, I guess the early to mid-90s, these were very difficult experiments, actually, to get LTP to last this long, or to have the slice last this long. And here, when they inhibit PKA, they wipe out the late phase of the LTP. When
Okay.
they inhibit CAMK2, they wipe out both the early and the late phase. And actually, I'll have to come back to this class because I always forget this is pertussis toxin.
Yes.
But I'm actually not sure what the point of this is. So,
Yeah, me neither.
or at least I think it's pertussis toxin.
Oh, god damn it.
And here you can see protein synthesis inhibitor reduces late phase LTP as well. So now we're going to switch from
I hate this
hippocampus to
so much.
cerebellum, and we're going to look at some of David Linden's experiments.
I don't care.
Using the same system that we talked about in the context of long-term depression. And again, this is long-term depression, but it's late phase long-term depression. Okay, so this is just to remind us of his system where he compared depolarization with ionophoresis of glutamate, and he has 2 sites. One site where the ionophoresis is paired with the depolarization, one site where it's not paired. The site that's not paired shows no LTD, while the site that's paired shows very nice LTD. If he applies a protein synthesis inhibitor, he gets LTD out to like 30, 40 minutes, but after that it begins to wane. And so this is without, this is the unstimulated or unpaired, and this is the paired with anhysamycin. So there's a protein synthesis dependent effect, certainly by the time you get out to 60 minutes. And basically, this is the same story, except he varies the time of the anhysamycin exposure. And if he adds the anhysamycin a half hour after pairing,
Make it back.
story, 80 minutes or unpaired, and this is the paired with anhysamycin. Out to like 30, 40 minutes, but after that it begins to wane.
When did he apply the drug?
System that we talked about in the context of long-term depression. And again, this is long-term depression, but it's late
Yeah, I know what
phase
long-term depression is.
Okay, so this is just to remind us of his system where he compared depolarization with ionophoresis of glutamate, and he has 2 sites. One site where the ionophoresis is paired with the depolarization, one site where it's not paired. The site that's not paired shows no LTD, while the site that's paired shows very nice LTD. If he applies a protein synthesis inhibitor, he gets LTD out to like 30, 40 minutes, but after that it begins to wane. And so this is without, this is the unstimulated or unpaired, and this is the paired with anhysamycin. So there's a protein synthesis dependent effect, certainly by the time you get out to 60 minutes.
Where did he put the drug on?
And minus of paired shows very nice LTD. So there's a protein synthesis dependent effect, certainly by the time paired with anhysamycin. So this is without, this is the unstimulated or unpaired, and this is the paired with anhysamycin.
Okay, forget it.
So
It's
there's
got
a
the
protein
drug.
synthesis dependent effect, certainly by the time you get out to 60 minutes.
Wait, did you say without
Out to
isomycin?
like 30, 40 minutes, but after that it begins to wane. And so this is without, this is the unstimulated or unpaired, and this is the paired with anhysamycin. So there's a protein.
Did you say without God
system that we
damn it.
talked about. We're going to look, or at least I think it's pertussis toxin. We're going to look at some of David Linden's experiments using the same system that we talked about in the context of long-term depression. And again, this is long-term depression, but it's late phase long-term depression. Okay, so this is just to remind us of his system where he compared depolarization with ionophoresis of glutamate, and he has two sites. One site where the ionophoresis is paired with the depolarization, one site where it's not paired. The site that's not paired shows no LTD, while the site that's paired shows very nice LTD. If he applies a protein synthesis inhibitor, he gets LTD out to like 30, 40 minutes, but after that it begins to wane. And so this is without, this is the unstimulated or unpaired, and this is the paired with antisomycin. So there's a protein synthesis dependent effect, certainly by the time you get out to 60 minutes.
Okay.
And basically, this is the same story, except he varies the time of the antisomycin exposure, and if he adds the antisomycin a half hour after pairing, it no longer affects the LTD out at 2 hours, which is pretty interesting. It's very similar to the Aplysia story. In other words, the protein synthesis is important at this early stage, but not subsequently. On the other hand, if he adds the antisomycin just a couple of minutes after the paired depolarization and glutamate anaphoresis, now by about 45 minutes, the LTD begins to affect. So in this experiment, he uses the more isolated system where he pulls a dendritic macropatch off the dendrite, and now he can pair depolarization with anaphoresis of glutamate. And what you can see here is, unlike in the previous slide where he had very stable LTD out to 2 hours, here in the control, the LTD begins to fade after an hour and then fades away. But interestingly, even in this isolated dendritic macropatch, you have an effect of antisomycin which shortens the duration of the LTD, suggesting that local protein synthesis in this dendritic macropatch is contributing to the LTD. So later, there's a contribution of transcription, or presumably, or proteins, perhaps new protein synthesis from the cell body. But at this early time, out to about 45, 50 minutes, the LTD is completely stable. But after that, If you I guess the right way to say it is,
explain
out to more than
it.
an hour, the proteins, the
Oh,
LTD
God.
is stable in the absence of any transcript or new protein synthesis from the cell body, and then it fades. But in this window of time, protein synthesis is important locally, so in the dendritic macropatch. He then, David Linden and his collaborators, including David Ginty, looked at a couple of different transcription factors using dominant negatives. So ATF2 is in the CREB family, and what they found was the dominant negative CREB blocks the LTD after about 45 minutes. And this is the whole cell now, of course, because otherwise they couldn't study the effects of transcription. And they express these in the Purkinje cells in culture. And the dominant negative ATF2 has no effect on the late phase LTD out to about 2 hours. And these are just control experiments to verify that calcium influx is not affected by these proteins that we're expressing. Okay, I want to go back to Aplysia for a little while, and
I don't.
although I'm not going to be talking about any detail,
Oh, good.
I just want to summarize a large number of experiments by Craig Bailey in which he and his collaborators found that with long-term sensitization, there's an increase in presynaptic processes from the presynaptic sensory neurons. So these are fine branches that contain presynaptic varicosities. And these are reconstructions from EM in the ganglion, but you can see, so these cells were filled and then reconstructed in EM. But then you can also see dramatic changes in presynaptic varicosities in culture. And we'll see that in some experiments in culture in a while. One of the things that Bailey and his colleagues observed was that if they looked at a cell adhesion molecule on the surface, and this is somewhat related to NCAM, which is neural cell adhesion molecule, and they call it APCAM or plesia cam, and it's in the same superfamily as fasciclin-2, the immunoglobulin superfamily. What they found was that
means nothing
with
to me.
serotonin, there's a reduction in the cell adhesion molecules on the surface of the APCAM on the surface, and so they look with immunogold, and what they see is that serotonin reduces the staining on the surface, and it's a protein-synthesis dependent reduction in APCAM staining. On the other hand, there's an increase in the staining in the cytoplasm. So they believe there's an endocytosis of the cell adhesion molecule. And how might that work? In this cartoon, they show their model, which is somewhat speculative.
I don't care about your model. When I was in London, somebody called me a racial slur for Middle Eastern people, and I don't know why, because I'm not that. I remember that being particularly strange. Somebody like shouted it out from a car that had pulled up. It's like, oh, hello. That happened. The end. That story didn't really go anywhere. I just felt like sharing. What are they talking about in this model?
In which
Dogs
the
are better.
homophilic association of APCAM
Homo.
on two different sensory neuron processes,
And what?
such as here, stabilizes the processes and impedes growth. And on the other hand, when they give serotonin, there's an endocytosis of this cell adhesion molecule.
Yeah, but I want my
which
dog to be
creates the
biased to me.
possibility for
I don't
new,
care if my dog likes you.
synaptic growth. And it turns out there's also an APCAM interaction with the postsynaptic long and composable synapses. Okay, that's where I'm going to stop, and we'll continue.
You have
this
far too many
discussion in
programs
class,
open, my man.
Talking about synapse specificity.
It's a good sample.
With fast, then plot, then have a set up a system where you could have a barrier and seal this gap with Vaseline, and then he could apply serotonin either to the peripheral. He have a
Why does he have 3 different versions of Adobe? Acrobat, 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 Flash Player Update, Apple Software Update, iMovie, no idea. I think that's just a disk he inserted? I don't remember. Print Job, something, Typey, Skype, don't know what that is, QuickTime, another iMovie, Mathematica, I don't know, whatever that is, no idea, Prism, Excel, Sherlock. I don't know why people can't use computers. I don't know why people with PhDs can't use computers. It's not hard. Like, Christ. Why is it always... Oh, I don't know how to use the projector. Like, no, yes you do. You know how to use a projector. Ugh, God. Now you get to listen to my printer. Like, Windows P. There, you did it. You're done. Go home. No. You're not beginning to understand it. Christ. It's 2016. How do you not know how to use a printer? Why do you have the PhD and you can't use a printer? When I can use a printer and you don't have a PhD. I mean, I could use a printer and I don't have a PhD. I got the stack of papers every time I have a lecture by this guy. It's just a stack of stuff and he grills you on every single article, every single figure. It's like, I don't know the answer. I'm sorry, I'm a moron. Alright, can I go home? I wonder how the person who makes Sonic X torture porn is doing. Somebody died. Do I know what you could eat for more intense or frequent dreams? No. Coffee, maybe? I always thought it was coffee because my thought was coffee prevented you from going into a sleep cycle that is incredibly deep and kept you on the cusp of rapid eye movement, and that would have helped. That's based on absolutely nothing. Antidepressants, maybe. Those don't work, though. Oh God, why am I reading this? The Sonic X torture porn lady. God. This is so painfully written. I swear to God, this person has got like a medical fetish, and it's the worst. She is still doing the same, yes. It is, like, the worst. Every single thing is written in miserable detail. Like, the nurse takes off his nasal cannula and puts the simple oxygen mask onto him at 6 LPM, parentheses, liters per minute. Why didn't you just write that? Or make it liters per minute, parentheses, LPM. Don't be like, oh, it's 6 LPM, or for you idiots, liters per minute. Like that. No. It's either you define the term with the abbreviation after so your reader knows what it means, or you don't use the abbreviation at all. Don't be like, it's this, but for you people, it means that. Like, no, stop. Good job. Good job. You know what LPM stands for. I'm proud of you. Thanks. Well done. I don't understand why, if you like a character so much, you just want to see him die in a hospital bed like every day. But I can get back at her through public shaming. Sorry. It's my only skill. Oh no, my stapler isn't working. Also, when people on the internet talk to each other is like pats your head tenderly, I hope you can make it to Cleveland, sweetie. It's like, yeah. Stop trying to be my mom. staple, you bastard. Oh, it did. Ugh. I wonder what happened to her dweeby boyfriend. I pick on children all the time. That's why I pick on you in the chat. Regenerative. I can't even read words. Observation. God, I love how it's like Dr. Robotnik just hugging this busted kid in the hospital. Like, why would you do this? What a weird thing. Also, he wrote a lot of poetry and they're not very good. More from DeviantArt. Oh. The lady who draws the Chris torture porn posted a selfie and I clicked on it and all of the related selfies from a different person are kind of cute. I guess that doesn't help her out much. A new meta. God, all these DeviantArt comment chains are the worst. Like, you don't have to respond to everything. Someone says nice, reply is thanks. Reply again is you're welcome. Oh my Christ. Then the internet boyfriend says, "Aw, sweetie, you are so adorable. Seeing your face is all I need to feel better. You are like an angel from the heavens whose warm smile can wash away my worries. Little Chrissy is in very good hands." Chrissy being the stuffed plush of the character that she draws being medically tortured every week. Ugh. Gag me. Christ. I should just have... Can they just make... Oh man, that is actually adorable. I want the second one to the right. But I'll take any of them. Can they just make a comment section plugin that like replaces the comments in comment sections with like actual smart comments? Like, it doesn't even matter what the content is, just better. Chris said with concern, "Doctor, what's wrong with me this time?" Eggman, taking a blue pair of nitrile which you misspelled gloves and begins putting them on. "You're in a state referred to as toxic fever. You're infected enough to kill an entire village of people." Oh my god. As the bacteria dies, it produces lethal amounts of cyanide in your bloodstream. In addition, you exhale cyanide gas. Alright. Why is he not dead? We'll need to get you the antidote for cyanide in your blood. He should be dead. Dead! Oh my god. Why are you writing this? Christ. I don't understand. Eggman's looking for something to slow the progression of the poisoning, finds a cap syringe of hydroxycobalamin. Aha! Comes over with it a butterfly needle, in parentheses, to draw blood with, a sealed test tube, in parentheses, to draw blood into, and an alcohol swab. Oh man, I want to hug that dog. This is so weird. Just as I assumed, you're anemic again, and you don't have... Or I'm sorry, you're gonna need a blood transfusion. Problem is, my facility doesn't have a definite blood bank yet. Because it's new, parts are under construction. Eggman says, would you consider taking my blood? Just let the kid die. Like, Jesus Christ. Every single week you torture him with some new horrific disease. Just let him go. What's Eggman's blood type? I don't know. I don't know. Oh, look at the dog. Is that real? Oh, man. You're killing me. God. This is so stupid. Wonderful dialogue, dear. You're getting very good at anatomy and color variety. God. Why? Why? Why are you doing this? Why do you write stories of a kid being killed in the hospital? God damn it. Oh my god. Oh my god. These comments. The turtles are so cute. I love turtles, though I love Sweetheart more. Such a lovely lady. In response to the girlfriend says, you know what the signature dish is? Turtle soup. My poor turtley friends. And then he makes a sad face, and then he makes a response. Don't worry, dear. Huggles. And then the next response is hugs. All I need is you, babe. Heart. Ugh. God. I like this one. Okay, be prepared. It might count as a jump scare. Like, what? Why are you injecting him in the brain? It doesn't even work that way. Like, what? What is happening in this image? Like, what? I don't understand why you keep making these. God. This person just has a fixation on everything medical, but not like in a useful way where they'll actually be a doctor. It's just in a weird Saw sort of way. My God. Oh, God. It's the worst. Somebody says, nice work, and then the response is, what do you like about it? Like, no, if someone compliments your thing, you don't put them on the spot and say, no, praise me better. Jeez. Ugh. Then she says, I passed out a similar way while ice skating. And then someone says, you passed out? And then she says, no, wait, I didn't pass out. I hit my head. I don't know what I'm asking. Seriously, why does this kid have a heart attack, walks a few steps, and then coughs up blood? Why? How does that even happen? Also, just put the kid down. Jesus. Like, he's not coming back. He's just dead. Oh yeah, and then there's also the character just called Minority-chan. I forgot about that. Mistress Minority. That's not... Why is Mistress Minority white? Oh, God. Somebody wrote, wow, poor Kurisu-chan. And then someone said, do you mean Kurisu-kun? Ugh. Ugh. God. Minority-chan. Chan. I don't even care. I hate all of it. Why? Why? Let the bloodbath commence. Dude, he's like a cartoon 12-year-old. Ah, Christ. What a thing. Also, they tried to write Sonic GXT in Japanese, but that's not what they wrote. Looks like Sonitsuwa. They got the first two parts of Sonic right, and then like the second part just turns into a mess. I don't know. I should brush my teeth, pop a thing, melatonin, send an email. But I mostly just want to watch the drawing of an anime 10-year-old get killed. No, I don't. Yes, I do. Oh, I might. The internet boyfriend says, "Aw, my pretty lady is always pretty, no matter how she looks." Oh my god. Gag me. I want to vomit everywhere. I'm already vomiting. Barf. Just barf everywhere. Is it just low-hanging fruit to just make fun of DeviantArt kids? And these are like kids in their 20s. Here's an emotional piece of poetry. Somebody left a response. "I don't like this poem. It was like putting handcuffs on and trying to do jumping jacks. You know me. I'm a rebel of words, and conforming to a fixed set of rules for the sake of rules seems like it is meant to stifle creativity. I admire your work at doing such a poem, but be free and beware others' rules." What does that even mean? Who cares? Like, it's a poem. This 24-year-old male left a comment and said, "I find it hard to judge this work objectively, since I absolutely hate the structure that you employed in its creation. As a poet who is mostly focused on communicating emotion myself, the structure seems stifling and unnecessarily limiting." In summation, oh, piss off. "I think that the ideas are interesting, and the story may well be too, but the rigid structure you can find it too misspelled made it the worst possible thing for a piece, comma, in my eyes, colon, boring." Wow. What an idiot. I would have enjoyed it more had your ideas been given greater leeway to fly free. Stop! Everyone is so pretentious. You wrote a poem and you put it on DeviantArt. Do you want a medal? Do you think you're qualified to be Super Mr. Art Critic? God. Like, ugh. Good job. You wrote some words and you put it in a website full of bad anime porn. Like, okay. No, you can't go both ways on that. I don't feel like defending my argument, so you're just gonna have to accept it. A calm echo utters the refrain, "It's only a dream." Angry yells drown it out and scold me for daring to dream. Rejected stories burn in the fireplace before me, not a single one published, leaving me with just the dream. My parents spit into the fire, telling me to leave. They will not care for a son who only wishes to dream. My computer sprouts wings and flies straight into the fire. The inferno gobbles it up, mocking my only dream. Pencils snap, and when I pick them up, my notebook morphs into dust. The world has turned against me simply because of a dream. The room melts away, jeering fire replaced with cruel cold. My stories and home are all gone, all because I dared to dream. What happened? Those who pass me by on the city street stop to chuckle. It's a sad sight, they say, a life lost on a hopeless dream. No change falls into my lap, no good fortune comes my way. I've plunged into rock bottom all because I chased a dream. This is still going. I. God. I think poetry on DeviantArt has got to be the worst in general. Somebody drew an anime. Yep, that's an anime, I guess. And then they wrote a lot of paragraphs saying you can do it. Okay. Thanks. I guess. Oh, this one's got mature content. Her ladyship's order. These people want the hug society never gave them. I threw my hug into the fire and said it was just a dream or something. Another poem for my love. Ew. Gross. This is a gift for my precious Ser. God. The response is, Tay Tay, you have me weeping tears of joy, hugs tenderly. Response to that, hugs tenderly, cries happily with you. Oh, God. Happy, sad, angry, pick any one of those three, and I'll still love you. Time just loves to fly, and we fly alongside it. Two graceful lovebirds. You may remember my blushing and stuttering during our first date. I still remember my fierce internal debate at the end of it. To hug or to kiss. My gut guided me to hug, but now I'd choose kiss. It's funny to think how me faving a drawing started this whole thing. Yeah, no, that sounds about right. Curiosity took me to your page, and there I learned of your pain. Depression is hard. I knew that even then it was. So I sent a note. I offered an ear, and you kindly returned one, no hesitation. Anime and games, two loves that don't even match. What? No, it's like the same thing. It's like a synonym. Oh my god. Even if we drop, I'll search for you in heaven, my precious angel. I want to throw up on everything. God damn it. In which way can I crush people's dreams the fastest? Oh dear. Oh dear. This. How old is this person? 21. 21. That's. Oh dear. That's, I'm not very good at drawing, but I can do slightly better. Just a little bit. But I'm sorry. I know we all start somewhere. I know. I know it's cruel. I was surprised. I can't help it. I know. I know I'm the worst person. I'm sorry. But wait, no, no one had a problem when I made fun of the poetry. No one's like, "Oh, he's just the beginning poet." See? You have to stop me making fun of everything, or you have to stop me from making fun of nothing. Poetry is for dull-ass dweebs. Oh, God. Search DeviantArt for poem. Don't hide the mature content. I want to see the pornography. Show me the porn poems, please. And thank you. I want the one most popular. That's not a poem, that's just a naked lady. I want the newest poem. No, I don't. These aren't even funny. I don't know if this is a real thing. I can't even read it. Does anybody get anything for that? Nobody commented on it. I mean, he left a post on 420 that said weed, and that's it. And his title was Happy Weed Day. I don't know if he's pissed. I'm assuming these are all fake. Relapses are broadcasted, and pain is something to be reigned, like an anarchy that repeats itself until the end of time itself. Rumors from high school hallways and Snapchat stories, people forget that the monster exists and the story comes as close as the curtain falls. I have no idea what you're talking about. I don't know who that is. Was that old Anderson Cooper? Why? I don't understand anything. Why is the description for this boy I die over some McNuggies? Is there context? For like any of that? Just took a picture of a dead bird and put it on DeviantArt. Great. That's alright. Oh, that's amazing. Oh, God. I feel like none of this is real. Like any of it. This person drew this and said, I have a problem, entitled Object Hands and Monotone. Excuse me, I embrace my handicap of being monotone. How dare you? I'm reclaiming that word. Why are the more from DeviantArt images just lesbians kissing and ladies showing their butts? That's the lesbians kissing one, and that's the ladies showing their underwear. Well, I'm gonna go to bed now.